Next, we will have our only online talk for this session, and we'll have Marcus Savarese from Helsinki in Finland that will talk about unreported GNE findings from the Helsinki resequencing analysis.
So my name is Marcus Savarese and I'm from Helsinki. I will provide an overview of unreported GNE findings from our resequencing analysis here in Helsinki. And what we are currently doing here. So we run different genetic tests. Basically, we have in use a large gene panel covering over 700 genes, and we have so far analyzed more or less 3,000 patients, myopathy patients with this large gene panel. And we have an increasing number of exon sequencing, currently more or less 800, of genome sequencing, over 100, and above all of RNA-seq analysis with almost 300 samples analyzed. And from our cohort, or let's say our cohort is mainly based on Finnish patients, but with an increasing number of non-Finnish patients, mainly European, but as you will see also from other countries and continents. So what did we find? Well, a few years ago, we found the first family with a GNE myopathy in our cohort, reported here in 2016, a Greek family, homozygous for this well-known missense variant. But after that, we expanded our study and we identified a number of other non-Finnish families from France, Portugal, Italy, Switzerland, Egypt, Spain, and again, more recently, a second family from Italy. All the variants here reported are already known, and we have a combination of homozygous variants and of compound heterozygosity. All these patients were referred to us for distal myopathy, mainly with remed vacuoles, which is basically the main core of, or the main interest of our lab in Helsinki. We have collected clinical data of actually few patients out of this cohort. We calculated we have something like 15 patients diagnosed right now, and these clinical features refer basically to 6 of them, with an average onset of 32.6, with first symptoms in the lower leg, with foot drop, for example, and a couple of patients with weakness also in upper limbs and in hands. For at least these patients, for whom we have clinical data, a loss of ambulation was not observed, and respiratory muscle involvement was not observed. A single patient presented with a cardiomyopathy, an hypertrophic cardiomyopathy, but again, without a clear genetic evaluation of the patients, we can't really make a connection with the GNE. And the creatine kinase was normal or mildly elevated. For few patients, we collected MRI, and here you can see the MRI of the first 2 patients from the Greek family. And the MRI shows in one patient extended, let's say, fat tissue replacement with sparing of the hamstring, and in other patients, a more evident involvement of the posterior compartment. We collected again for a few patients, and here the sample is from the same 2 patients, muscle biopsies with remed vacuoles and with aggregation, in line with our expectation, because I said we mainly work on these forms, and thereby this is sort of bias. Whatever we collect, of course, in terms of DNA, they are from patients with distal myopathies and mainly remed on non-remed vacuoles. I want just to focus now on a very interesting finding from 2022, so a couple of years ago, when we started a screening of a cohort from Egypt. We had 2 families homozygous for the methionine threonine missense change, but then we have in a third family a single missense variance in heterozygosity. When we contact back the referring clinician, the diagnosis of GNE was, of course, one of the possibility, and we were missing thereby the second variant. We implemented the algorithm for the identification of copy number variants using our gene panel, and we define the exon 2, previously reported honestly exon 2 deletion in this patient. We confirmed the finding by the ReseGH. We have in Helsinki a ReseGH covering all the exons of all the myopathy causative genes. Thereby, we have an array with high resolution able to confirm even a single exon deletion. And this is an interesting aspect because, as already honestly discussed in literature, in the presence of a single GNE variant, causative variant, we should consider the possibility that there are intragenic deletions of the applications, and at least in our experience with NGS data, the deletions and applications of a single exon may be tricky. We detected the exon 2 deletions because we have a very high depth in our gene panel data, but honestly, I'm not so sure that we would have been able to see through an exome or genome sequencing with a low coverage the same deletion. In conclusion, we have 13 families, 15 patients identified so far, no finished patient in line with data that Professor Luckmuller has shown. Basically, causative GNE variants in the Finnish population are extremely rare. We are currently reanalyzing also unsolved cases wherever there is a GNE heterozygous variant, and whenever we have an indication that the GNE heterozygous variant may be pathogenic, we are currently trying to see the copy number variant data and spot possible intragenic small deletions of the applications. And concerning this specific cohort, we are still working on a data acquisition in order to draft a manuscript. Most of the work has been carried out by Mridul Johari, who is currently in Australia, and of course, Professor Rood has been the one collecting all the material and reviewing current the clinical data. And all these patients, as said, are from European or non-European countries. So a special thank goes to our collaborators worldwide. Thank you.